Hi everyone, this is Vignesh from VNMA. Today we are going to see about physiology of insulin. So insulin, everyone know about this word because insulin is a main factor or cause for diabetes mellitus. So today topic we are going to cover structure, synthesis, secretion, mechanism, effects, clinical aspects and treatment. So the effects, clinical aspects, treatment. So we are going to see detail in internal medicine, pathophysiology and pharmacology. So today we are going to see the basic physiology of insulin. So before going to this structure, we'll start with structure. Before going to the structure, so the overall both the insulin. So the insulin which is secreted from the pancreas. In the pancreas we have a beta cells. In a beta cells we have islets of Langerhans. From this insulin is produced. So when we see in the stomach, after we eat, will digest, will produce amino acids, fats and glucose. So these things will be absorbed in blood and it will go to the organs. So when the amino acid is absorbed in the blood, the insulin will use as a enzyme and then it will enter into the liver. So in a liver, this amino acid will start protein synthesis and then it will store in a muscle and then it will be used for another metabolisms. So and then the fat will converted into fatty acids and then it will be stored in skeletal muscles. After that, this glucose will absorb in the blood and then by the help of insulin, this whole process is done by help of insulin. It will go to the liver and then it will start a cycle called glycolysis and it will produce some energy and it used in a metabolism and then it will convert it into glycogen and then there will be a low density lipoprotein. So by the metabolism we'll get a products. The products will stored in adipose tissue and then some of them will used in other metabolisms. So this is overall about the insulin. So now we'll see the structure. So how the insulin will look like. So insulin which has two polypeptide chains, one is alpha chain, one is beta chain, which is joined by sulfide bond. So disulfide bonds, two polypeptide chains, alpha and beta chains, which is connected by two disulfide bonds. Disulfide bonds. After that, synthesis. This synthesis process in the pancreas, in the beta cells, in islets of Langerhans. So it will start from ribosome. Ribosome translate a specific protein for insulin, which is called as insulin mRNA. The specific protein for insulin. So this synthesis process, which is encoded by a gene called INS gene. this insulin mRNA, which is called as preproinsulin. So the insulin mRNA will look like, three chains, A, B, C, and then the signal sequence, this ribosome translated insulin mRNA, the specific protein, this signal sequence will attach to the A chain. This thing is called as peptides, A peptides, B peptides, and C peptides. This preproinsulin. This preproinsulin will enter into the endoplasmic reticulum. There will be a breakdown of this signal sequence. So there will be a preinsulin. After that, this preinsulin will enter into the Golgi bodies. In a Golgi bodies, there will be a breakdown of this c-peptide or a C chain. There will be a production of insulin. So this insulin will stored in a vesicles in a beta cells or in a pancreas. So insulin will stored in a this insulin will stored in a vesicles, small vesicles. So now how it will secreted from the pancreas, from the beta cells of pancreas. Secretion. So we have to know the three channels. This channel is called as GLUT2 channel for entering a glucose, and then we have potassium ATPase channel, and then we have voltage gated calcium channel for entering calcium. So GLUT2 channel, potassium ATPase channel, and then voltage gated calcium channel. So the glucose is absorbed in blood and it will go to the pancreas. So the glucose is entered into the cell via GLUT2 channel. When the glucose is entered into the cell by the process of respiration, there will be a production of ATP. So this ATP will affect this potassium ATPase channel. So there will be a depolarization potassium ATPase. There will be a depolarization in the potassium channel. So it will be sensitive to the ATP. So it will attack this potassium ATPase channel. So there will be a outflux of potassium and ATP. So by the depolarization, it will initiate the voltage gated calcium channel. So from the outside of the cell, the calcium will enter into the beta cells. The influx of calcium. So this calcium will attack the vesicles which present insulin. So this calcium will attack the vesicles. There is a presence of insulin. So this insulin, by the process of margination, will go to the cell surface, will go to the plasma membrane, and by the process of exocytosis, this insulin will pushed out from the cells. After that, how it is secreted out of the cell by the process of pulsating pattern. What is meant by pulsating pattern? By the slow process which is used after we eat, and then by high secretion, longer time secretion is used for cell growth, cell metabolism, cell developments. So by the margination, exocytosis, pulsating pattern, and then longer secretion. So GLUT2 channel, KT, potassium ATPase channel, and then voltage gated calcium channel. So now we'll see the mechanism of insulin. Mechanism. In a tissue surface, there will be a insulin receptors, and then there will be a GLUT4 channel. So when the insulin will come to this insulin receptors, it will initiate the cell nucleus. This is a tissue cell. This cell nucleus will initiate the GLUT4 channel to open the channel, so the glucose will enter into the channel. There will be an increasing uptake of glucose, the energy. So now we'll see the insulin receptors. So the insulin receptors, which has two chains. One is alpha chain, one is a beta chain. Will exactly look like this. Under the beta chain, there will be a tyrosine kinase. In the tyrosine kinase, there is a presence of phosphate. When the phosphate is attached to the tyrosine kinase, it will activate. So when the insulin is come to the insulin receptors, it is diagnosed by alpha chain. This alpha chain will fold the insulin, will cover the insulin. After that, it will go downwards, and then this beta also fold the insulin. So when there is a covering and folding, so this phosphate will attach to the tyrosine kinase. So there will be activation of phosphorylation cascade. This phosphorylation cascade will initiate the protein channel, which is GLUT4 channel. So this GLUT4 channel will open up, and then the intake of glucose will be higher. So the insulin will come to the alpha chain, the alpha chain will fold, and then beta chain will fold, and then the phosphate will attach to the tyrosine kinase. There will be activation of phosphorylation cascade. This phosphorylation cascade will initiate the protein channel, which is called as GLUT4 channel. So the GLUT4 channel will open up, then the glucose uptake into the cell will be higher. These insulin receptors will present in the cell surfaces. So now we'll see how it affects on the livers, adipose tissue, and then muscles. Effects in the liver and muscles. There will be an increase of glucogenesis, and then will be decrease of glucogenolysis, and then in the adipose tissue there will be a decrease of glucogenesis, and there will be a lipolysis in the adipose tissue, and there will be a increased amino acids uptake in the liver, muscles and adipose tissue. And in the liver there will be a increased glycolysis. So in the liver and muscle there will be increased glycogenesis, decreased glucogenolysis. In the adipose tissue there will be a decreased glucogenesis, and in adipose tissue there will be a decreased lipolysis. There will be a increased amino acids uptake in the liver, muscle and adipose tissue. In the liver there will be increased glycolysis. So how we can stimulate the insulin? Stimulate by the gastrointestinal tract hormones and then acetylcholine. How we can inhibit the acetylcholines? How we can inhibit the insulin? Adrenaline and then noradrenaline. So and then the clinical aspects which are related to the insulin, mainly the diabetes mellitus type 1 and then type 2, which is insulin insufficiency, which is insulin resistance, and then we have other types of diabetes mellitus called gestational diabetes mellitus, and then we have insuloma, which is a tumor in the beta cells in the pancreas, a tumor in the beta cells, and then metabolic syndrome, which is later on converted into type 2 diabetes mellitus, and then PCOD in the female, polycystic ovarian disease or syndrome, so later on converted into type 2 diabetes mellitus. So what are the treatment for the diabetes mellitus? For insulin insufficiency for type 1 will give subcutaneous insulin injections. For the type 2 diabetes mellitus will give metformin. This is the basic thing about the insulin treatment. So later on we'll see about the treatment in the pharmacology and internal medicine and pathophysiology, and then mostly we'll tell the diets. So I think you cleared about the basic physiology of insulin. So this is all about the insulin. So thanks for watching. We'll continue to some other topics on some other videos. Thanks.